Right. When the Earth was formed 4.5 billion years ago, magnetic fields were already present. And they had been since the Big Bang, 9 billion years earlier. 2 billion years ago, aquatic magnetotactic bacteria evolved, which contain a chain of magnetite in their bodies, enabling them to swim along magnetic field lines of the Earth to find food. 90 million years ago, the avian magnetic compass developed, enabling pigeons, for example, to detect magnetic field changes as low as 10 nanotesla, which they use for navigation. 3 million years ago, modern man evolved, some of whom appear sensitive to tiny fluctuations in the geomagnetic field due to solar storms. So by the time electrification was introduced in the UK in 1878, it was already the case that all species of living beings could sense these fields, and the hint that there may be adverse health effects. Now I'm going to talk mainly but not exclusively about power frequency and magnetic fields and health. Power frequency. I'm very interested in candidate mechanistic pathways. I'm a physicist by background, which is a way of saying I'm not a biologist. So I hope I know my limitations in that regard. So I'm interested in the primary interaction between physics and biology. Let's go back to geomagnetic storms. These originate from events from the sun, and there are around 4 to 5 such events per year of interest. Those giving ripples of magnetic fields of a mere 100 nanotesla, lasting for 1 to 5 days. And there's quite a literature showing various acute human health effects associated with geomagnetic storms, affecting a proportion of the population. Even pigeon migration is disturbed by geomagnetic storms. There's a word for this phenomenon of these health effects. The word is called electrosensitivity. Many people ask, how can such small changes in magnetic fields possibly have any health effects against the static field of the Earth, which is 50 microteslas, much, much bigger? And the answer is that it's the change in field that the body detects. And it isn't a physics response, it isn't just a biological response, it's a behavioural response, a learned response. Imagine you have a tiny itch. The pain involved in that itch is probably very, very low, but it can be so annoying and so stressful if it's persistent. I put this slide in just to show you some of the papers on health effects of geomagnetic storms. Let's move on to power frequency and magnetic fields now. Average levels in the home are very low, around 0.05 microtesla. But if you live under a power line or near to a power line, levels can be tens of microtesla or several microtesla. There are high levels associated with appliances like hairdryers, but we don't use those all the time. Crucially, there is a doubling of childhood leukemia risk associated with average exposures above 0.3 or 0.4 microtesla. And more recent meta-analyses of the epidemiology have shown a 30% increase above 0.2 microtesla. Magnetic fields are classed by IARC as a 2B possible carcinogen. We've heard that this morning already. Just to go over that in slightly more detail, the IARC 2B classification was in 2002, based on the pooled analyses of Olbom and Greenland. We have this update from Zhao et al. 2014, where for the common acute lymphoblastic leukemia in children, the excess risk of 0.4 microtesla compared to the referent is 2.43, and above 0.2 microtesla is 1.31. But there are many, much more prevalent adverse health effects associated with power frequency magnetic fields. Adult leukemia, adult brain tumours, male and female breast cancer, depression, suicide, heart disease, Alzheimer's disease, miscarriage of motor neuron disease. If I may quote one of our papers, Michael Carroll and I in 2008, we looked at the aggregated p-values for adult leukemia and adult brain cancer. And the p-value for the association for both of these endpoints was off scale. It was less than 10 to the minus 7. Only a 1 in 10 million chance that the that the association occurred by chance. Now, p-values are sometimes treated with skepticism, so let me just mention that p-values are used in science in general, and in 2013 the Nobel Prize for Physics was awarded for the discovery, in inverted commas, of the Higgs boson. Now it's in inverted commas, the word discovery, because discovery is defined in particle physics of a p-value less than 10 to the minus 7, i.e. what we found, i.e. 99.59% proof the association doesn't occur by chance. We are, ladies and gentlemen, still waiting for our Nobel Prize. It hasn't materialised just yet. Now, one of the key mechanisms I want to talk about today is how EMF, magnetic fields, affects melatonin. Melatonin is a key component of circadian rhythms. Melatonin is produced in the pineal gland at night when light levels fall below about 200 lux. Melatonin is a powerful natural antioxidant and anti-cancer agent. If you are exposed to light at night, this can fully suppress nocturnal melatonin. Magnetic fields also suppress melatonin, typically by 7 to 14%, a lower amount, but they do suppress melatonin. Richard Stevens in 1987 proposed that exposure to light at night in EMF may increase the risk of breast cancer in night shift workers by melatonin disruption, more generally circadian rhythm disruption. The rest is history, and you can read all about that in the IOP 2010. They classified night shift workers as a probable carcinogen. Now, there's a lot more I could say about melatonin, and I would refer you to the talks at our recent Childhood Cancer 2018 conference. The videos of all the talks are available there for you to watch. Now this slide looks a little bit busy, but it is to just try to illustrate just how much has been done on magnetic field disruption of melatonin, and to introduce this idea of cryptochromes. Magnetic fields have been shown to disrupt the action of individual pineal cells in the pineal glands. They've been shown to suppress melatonin in animals, especially cattle, in the real outdoor situation near power lines. And especially in humans, particularly in humans, down to 0.2 microtesla, a review here. But, as I said earlier, melatonin is part of our circadian rhythms, and they are controlled by a series of genes called the clock genes. And the most important one from our point of view are the cry genes, which express the cryptochrome protein molecule. And it's the cryptochrome protein molecule in the eye, which is in the regulation of circadian rhythms. We're going to come on to another application of cryptochrome in a moment. But for the time being, we have this is now historic theory of one possible pathway, that magnetic field exposure disrupts melatonin, leads to increased cancer risk. Now, this model has been developed considerably in recent years. And we have a very high degree of natural thinking entering the equation here. So I'm going to move absolutely lateral
and I'm going to ask you, what have these species, and many more which I could put on here, what have they got in common? And the answer is their remarkable sensitivity to magnetic fields, notably used in migration, in migratory animals like loggerhead turtles and birds. Birds such as homing pigeons, I've mentioned already, have an estimated detection threshold actually below 10 nanotesla in the latest experiments. So, how do migratory animals navigate? Well, if you're going to navigate across the Earth, you have to have two measurements. Humans, if they don't have a sat-nav, will use latitude and longitude. Animals use the magnetic field of the Earth, and their two measurements are the intensity of the field and the compass direction. The intensity is measured by magnetic particles in the body, and the orientation by a chemical compass in the eye. Now I'm going to skip over magnetic particles in animals and to explain why they're interesting to humans. And they are interesting to humans because the human brain contains magnetic particles in some abundance. Some of these are huge, 600 nanometer chunks, 0.6 of a micron. And these are big enough to physically vibrate in a 50 Hertz 0.4 microtesla field and a radio frequency field, and so they will put mechanical stress on neighbouring cells. I'm going to skip over ferritin from the time being. But it's this second mechanism which has really caught the imagination amongst physicists and physical chemists. It relies on a process called the radical pair mechanism. Low intensity magnetic fields can increase the lifetime of free radical pairs. So you create a pair of free radicals. Much talked about in animals is they're created by light, a light photon. And you increase their lifetime from initially 10 nanoseconds up to a microsecond by the action of magnetic fields. This means that the longer-lived, when they're longer-lived, they undergo different chemical reactions, and that's used as a signal in the chemical magnetic compass. But in living longer, these radicals become more available to cause biological damage. That's what's of interest to ourselves here. Now, Ritz et al. In 2000 suggested that the avian compass operated in cryptochromes in the eye. So I've got a couple of slides on Ritz's work. Thorsten Ritz in California, where I was at the time, in Berkeley. Ritz et al. 2000 proposed that the avian magnetic compass worked by the action of the radical pair mechanisms on cryptochromes in the eye. And remember, I can't say often enough, cryptochromes are best known for controlling circadian rhythms. So here's a schematic of a cryptochrome. The idea was that light was needed to create the radical pairs, and as the bird's head moved, spanning different intensities of magnetic field as it tried to align itself in the Earth's field, you got this change in reaction products, which were used as a signal. But what Thorsten Ritz postulated was, well, if this takes about a microsecond to occur, can we interfere with this by exposing the bird to the inverse of a microsecond, and that is a radio frequency EMF of a megahertz. And the rest is history, because in a very nice Nature paper in 2004, using only 85 nanoteslas, Ritz et al. Found that broadband 0.1 to 10 megahertz or single frequency 7 megahertz fields did indeed prevent the birds from orientating in the Earth's field. Now, the next slide is quite revealing. You see, there was a group, a research group at the University of Oldenburg, North Germany, who were trying to reproduce these orientation experiments where the robins, in this case, European robins, would orientate in the spring migration in a particular direction. But they couldn't get them to orientate at all. And until someone pointed out that there were a number of radio transmitters nearby. And look at we're talking up to 5 megahertz here. We're only talking maximum 10 nanoteslas. Tiny field. So these were in wooden huts, these birds. So they were unscreened. So someone suggested you put the birds in electrically grounded aluminium screen huts, and lo and behold, the birds could orientate. Now this experiment took 7 years to get to the stage of a Nature paper, because obviously people were sceptical that background EMF from radio transmitters could possibly disturb poor bird orientation. But there you are, that is now a robust finding. So, let's get down to the nitty-gritty. Are human cryptochromes magnetosensitive? 10 minutes to go. Right, okay.
Don't worry if you run over slightly.
Well, okay. Well, the answer is yes. This experiment used Drosophila, fruit flies. Drosophila melanogaster, fruit flies. These fruit flies are magnetosensitive, but the first thing the group did was to knock out the cry genes, and then they were not magnetosensitive. So that proves something. No cry genes, no cryptochromes, no magnetosensitive. And they were admittedly using quite high fields here, but then the group introduced human cry genes into the fruit flies, and lo and behold, this rescued, the term they use, rescued their magnetosensitivity. So cryptochromes from the like of me and you and Erica were able to rescue these poor fruit flies that couldn't orientate, okay? Because they had no cryptochromes. Right. So, are human cryptochromes a universal magnetosensor key to health effects? Well, the avian magnetic compass requires light, blue light, to create the necessary radical pairs. So the compass is in the eye. But cryptochromes are found in all cells and tissues. So if they were magnetosensitive by the RPM, we can have all of our cryptochromes creating biological damage by magnetic fields. For that to happen, the radical pair mechanism would have to operate without light, because most of our cryptochromes are in our bodies. So I want to just take you through a few steps here. Radical pair mechanism, cryptochromes, the dark, and the word I'm sure you've heard so much about before, reactive oxygen species. Right, the first paper, lead author Roswitha Wilczko in Frankfurt. One of the co-authors, Margaret Armad. Now I'm singling out these papers with Margaret Armad as a co-author because Margaret Armad at the University of Paris is credited with the discovery of cryptochromes in the first place. Okay? Title: Light-dependent magnetoreception in birds, crucial step occurs in the dark. Poom and Armad, this time in the plant, Arabidopsis thaliana. Magnetosensitivity occurs during flavine reoxidation in the dark. Now, in case you're thinking that we're about to overturn all this light dependence in the bird compass, that's not what's happening. The thoughts are there is another radical pair operating in the cryptochromes. There's another one. And the next paper is the one really of interest, only just out, September. I have to read the title carefully. Low intensity electromagnetic fields induce human cryptochromes. I need to explain, these are our fruit flies again. These fruit flies with the human cryptochromes in, they are a model system for human cryptochromes. And I'm calling them EMFs, or the authors are, because these are 10 hertz pulsed EMFs. They're not oscillating, they're not sinusoidal fields. Induce human cryptochromes to modulate interstellar reactive oxygen species. What does that mean? We conclude that exposure to weak magnetic fields induces the production of reactive oxygen species, not the release of them as though they were trapped in there. We talk about release of ROS, the production of them, actual synthesis of them in human cells, and that this process requires the presence of cryptochromes. So I looked at how many papers there were on the issue of magnetic fields and reactive oxygen species and DNA damage, etc. And I think there are at least 200 out there. Martin may well, yes, he's nodding in agreement. But what's different is that Sherrod et al. Has shown you've got to have, in this fly system, have got to have cryptochromes present. So, well, it's only one paper for the moment. To answer the question, are cryptochromes the magnetoreceptor or a key magnetoreceptor? I haven't quite finished yet. I've got a couple of minutes to go. Yeah. I've got something else to show you. Going lateral again. Genomic instability. Now, this is a hallmark of cancer. Let's go back to 1992 with an ionizing radiation experiment. These are hematopoietic cells, and they're exposed to actually alpha particles from plutonium, doing a classic radiobiology experiment. Expose the cells, arrest the cells in beta phase, i.e. the first cell division, and look for chromosome damage. If there is damage, the answer is yes, and if there's no damage, the answer is no damage, end of experiment. And that sums up 50 years of radiobiology. But Manura Kardim et al., they were smarter than that. They waited 10 to 15 cell divisions, and then, out of the blue, spontaneous, miscellaneous chromosome damage of all sorts that we're used to seeing. Genomic instability appears many cell divisions after the original exposure. Now, this has since been seen in metals and chemicals and known carcinogens. You know what's coming next? Guess the next slide. Now seen with magnetic fields. Magnetic fields were demonstrated by Jukka Juuta-Leiden's lab. This is Jukka Lukonen, the other Jukka in Finland. They have demonstrated that magnetic fields engender genomic instability in exactly this way. Ladies and gentlemen, magnetic fields behave like any other carcinogen. Period. Okay? So let's put things together. I've repeated this so many times I won't repeat these bits. Here's the model. So this is the new model, and this is from Jukka Juuta-Leiden. It came out in August. Magnetocarcinogenesis. Is there a mechanism? So magnetic field exposures act on cryptochromes via the radical pair reactions, leading to circadian rhythm disturbance. This is all via the eye. But throughout the body, ROS production, DNA damage, and calcium efflux. Can I add that to the list? No? Okay. He'll tell me in a minute.
It's influx, not efflux.
Okay, so okay. I told you I wasn't a biologist. Leading to genomic instability and leading to cancer. So that's the new model. Don't forget, that the epidemiology of ELF, if we take into account all health effects, is highly robust. This isn't going to go away. And also, can I again mention magnetic particles, because they may also feature in this story. So thank you very much, and have a good bonfire night.